Okay, guys, we're going to go through the contraction mechanism, the actual proteins that are doing the interacting, and also talk about some energy for contraction. So, for the skeletal muscle contraction, what you're looking at is an animation of a sarcomere, where the actin and myosin filaments are basically sliding past each other, which is why it gets the name of sliding filament theory. And we'll go over the specifics uh, of all these things. So here are, if you look at this previous one, the really dark, the thicker blue line are the myosin filaments, which are thicker. They're all just like this bundle of wires, basically, and each little wire has a little head at the end. And so there's a bunch of these myosin heads per myosin band, myofilament band. And so um, that's the part that's going to attach to the actin and do the actual pulling, the contracting parts. And so on this picture, you have the other three proteins that are important. The actin and tropomyosin are sort of twisted together. There's sort of two strands here of actin, the blue spheres on here. You also have this like green sort of rope called the tropomyosin, and the kind of spaced out every once in a while troponins. The actin and tropomyosin are like ropes kind of strung together. Troponins are just sort of scattered along this big long chain of actin, which you can see are, you can't see, like one of these fibers or myofibrils here would be about as thick as like one of these, all of these guys together. So you can see the difference in thickness between myosin and actin with the other two proteins on there, which is why on this animation and a lot of the ones you see, the actin filaments, which are the red ones, along with troponin and tropomyosin in there that you just can't see, are really thin, and the myosin are really thick. So these are, and the troponins are the ones that are getting that calcium that we saw in the stimulus for contraction stuff. They're the ones that are basically holding the velvet rope, which is the tropomyosin, which when they get that calcium, they change shape, which causes this whole thing sort of to twist, and that exposes the myosin, actin myosin binding sites, which currently in this picture are blocked. And so there you see an animation of all these parts working together. The myosin head here, which is pulling on the actin filaments. When the troponin gets out of the way, troponin's right here. And the tropomyosin is the blue sort of rope-looking protein. The actin are the orange dots in this animation here. So that's how the muscle contraction works at the most microscopic level that we'll talk about. Which, again, here's all the reasons why those proteins are interacting, is because there's a change in voltage, which is going to eventually get sent to those proteins by basically the signal is calcium. That's what's going to activate these guys to do their thing. And so there's the details of the contraction. As long as there's calcium in the system, these guys will continue to interact. One of the things that I've mentioned that it's kind of tough to imagine how the ATP causes this guy to actually let go. So the energized version of the myosin head, as long as the binding site is available, the energized version of the myosin head will come up, make a chemical bond briefly, and then since it's a protein, it changes shape like we've seen a bunch of times. It changes shape, and that's the bending and pulling that you saw. So the ATP is actually causing it to release release the myosin head from the actin filaments. And so there you can see the action of the myosin head releasing because it's energized. There's energy in the system because it's a muscle that is most likely got energy available to it because the cell's still alive and it's still making energy. So here's the rest of the contraction stuff that was already mentioned in the animation, including the enzyme that's going to destroy the neurotransmitter, causing that signal to stop being sent. And there are some things that you have access to that you can check them out if you want. there's kind of a sad little picture, but this makes the point of the ATP that's necessary to let go. So once the myosin heads lock in, bend, and pull, they cannot let go. The myosin heads don't let go, and so the muscles are stuck in contraction mode, which is why a dead body is called a stiff, because all the muscles have stiffened because there's no energy to allow them to relax. So here's the rigor mortis stuff. And if you can answer these questions, then you've been paying attention. I'm not going to go through them, but you can, of course, always ask them at any time. Here's the energy stuff. There are 3 ways to get energy inside of our bodies. There's basically 2 molecules, creatine phosphate or glycogen. And how that glycogen is broken down, which you probably learned about in bio class, aerobically, where there is cellular respiration taking place in the presence of oxygen, and anaerobically, which is, of course, without oxygen. So here's basically creatine phosphate provides phosphates, okay, because ATP is a triphosphate molecule. So when ATP gets used, it becomes a diphosphate molecule. And so now the phosphate from creatine phosphate can provide more phosphates for triphosphate to be made again. It's that simple. Here's all the cellular respiration stuff that you may be familiar with from bio class. Myoglobin is simply the oxygen carrying protein, but since it's talking about the relationship to muscles, it's called myoglobin. Myo is for muscles. And the oxygen can run out, but it's like always present. It's just a matter of the system trying to keep up with how much oxygen is in the system. Oftentimes you'll see people, like football games, when they're playing in Denver, these guys are sucking on oxygen masks. It's really not doing a whole lot. Their body has the oxygen, but it just can't access enough of it to produce the energy that it needs to keep going. Anyways, for anaerobic respiration, when there's not oxygen present, our muscle cells are doing lactic acid fermentation, which is a process that only creates really 2 ATP, which is certainly not the best situation, like in the mid-30s, if there is oxygen present, but 2 is better than nothing. So that's why lactic acid takes place. Lactic acid fermentation takes place, basically to keep those muscles going as much as possible. And a little bit of energy is certainly better than no energy. And so that buildup of lactic acid is what people experience when their muscles are sore, because those tissues have been kind of soaked in acidic solution, basically. So here's the energy use for a 100-meter dash for all you track people out there, all the big fans out there. That's what's happening. And then the muscle fatigue stuff will start in the next video.